Okay, welcome everybody. Thank you for being here with us today on behalf of Thrombosis Canada. My name is Lana Castellucci, and I'm pleased to share this evening's webinar with Dr. Peter Gross. I'm a thrombosis physician that's based in Ottawa, and I'm also a general internist. And Dr. Gross is based at McMaster University in Hamilton. For this evening's presentation, we're going to be talking about biosimilars. Is imitation the sincerest form of clinical efficacy? We have a great webinar set up for this evening, and biosimilars are becoming more commonplace in medicine. So we thought this was a really timely opportunity to discuss biosimilars as they apply to low molecular weight heparins, which is something that we all use in everyday practice. For the purposes of our webinar today, we would ask that you put your questions or comments in the chat feature. The Q&A has been disabled. So any questions or comments, please enter them in the chat, and we will aim to answer the questions and address comments at the end of the presentation. So our scientific planning committee is based on several of us who have different specialties. So Dr. Gross and I are thrombosis specialists. And we also had Ryan Kersner, who's a pharmacist in Montreal, and Dr. Moodley, who's a hematology oncologist in Saskatoon, participate in organizing this webinar and the information that we've put together for you today. We do have some disclosures to share with you, and I'll just let you look at this slide for a moment. And some other disclosures that we need to share. So in terms of financial support, the program has received financial support from Valio in the form of an unrestricted educational grant. This program has not received any in-kind support. There are potential conflicts of interest. So for myself and Dr. Gross, we are eligible for an honorarium from Thrombosis Canada, and Valio may benefit from the sale of a product that may be discussed during this program. Additional housekeeping matters. So to mitigate potential bias, the content of the program was developed by the planning committee and peer reviewed by Thrombosis Canada. Recommendations involving clinical medicine are based on evidence that is accepted within the profession, and all scientific research referred to, reported, or used in the program in support or justification of patient care recommendations conforms to the generally accepted standards. And for accreditation purposes, this event has been accredited under Section 1 of the MOCOMP under Royal College of Physicians and Surgeons of Canada, and was approved by the University of Ottawa's Office of Continuing Professional Development. And participants can claim up to 1 hour, and the credits are automatically calculated for you. So we have several learning objectives for our webinar today. And at the end of the program, we hope that our participants will be better able to describe at a high level how evidence for biosimilars is assessed by regulatory agencies in Canada, to discuss criteria in evaluating and choosing biosimilar low molecular weight heparin agents, and to provide effective education on biosimilars to patients and care providers. So we'll start with an overview and some definitions of biosimilars. And we'll begin with a question. So question 1, I'll just read it out here. Which of the following characteristics apply to biosimilar drugs? A, large complex molecules. B, contain the identical medicinal ingredients as the original brand drugs. C, made from living organisms or from their cells rather than chemicals. And D, manufacturing involves intricate biotechnological processes. And so I won't ask anybody to enter their information into the chat, but we will move on, and throughout our presentation, we'll touch upon several of these characteristics. And for the purposes of answering the question, A, C, and D are all correct. So biosimilars are not identical to biologics, and they must be shown to be highly similar without any clinically meaningful difference in terms of safety and efficacy between them to be approved by Health Canada. And so that's a key component here. They are not identical to their biologic origin drug. So understanding biosimilars, we're looking at biologic medicines that have revolutionized treatment in many areas of medicine that are disabling or life-threatening. And a biosimilar is a biologic drug that is proven to be highly similar to the branded biologic drug. Biosimilars are manufactured to the same regulatory standards with respect to safety, efficacy, and quality as the branded biologic drugs. So they are held to the same criteria as the original biologic drugs. And there are several definitions that I'm going to go through so that we're all having a common understanding of the terminology that we're going to use throughout the presentation. And so the terms that we're going to refer to are listed in the first column on the left-hand side. And we'll begin by defining biologic. And it is a large complex molecule that is made from living organisms or from their cells. And they're naturally variable because they're made from living organisms or their cells. The original licensed biologic medicine to which a biosimilar is compared, and it can be referred to as the reference drug or the originator drug. And many examples are listed in the far right-hand column. So enoxaparin is very relevant for us for today's webinar. Rituximab, as we all know, is a very common biologic agent, etanercept, and they're used for many different conditions that you can see here: ulcerative colitis, rheumatoid arthritis, psoriatic arthritis. So many different indications. And then we'll go on to the second term, which is biosimilar. So this is a version of a branded biologic product, which is similar in terms of its quality, safety, and efficacy to the originator biologic, but is not identical. And examples, again, are listed on the right-hand side. And these are all examples of drugs that are available in Canada and are biosimilar to enoxaparin sodium. And then you may be familiar with the last one here, BioBetter. So this is an original biologic molecule that has had improvements made to it. And it almost always precedes biosimilars, and it may have patent protection. And there are some examples, again, listed here. So BioBetters of rituximab as an example. And then we'll also look at chemical drug and generic drug. So a chemical drug is a small molecule that is created synthetically, and it has a simple molecular structure that's easy to copy. And a common example would be aspirin, which is, as you know, trademarked. And then the generic drug is, again, a small molecule that is synthesized chemically, and it's identical medicinal ingredients to the reference drug. And ASA would be an example of that. And so this is the terminology that we will be using throughout the presentation. And so we wanted to make sure that you have an understanding of what these terms mean. So when we biosimilars are not talking about generic medications. They are not the same thing. Generic drugs are small molecules that are easily synthesized chemically, and they contain identical medicinal ingredients as their reference product. But as we learned a few slides ago, biosimilars are not identical to their reference drugs. And biosimilars and their reference biologic drugs are large molecules. They're not small molecules. So they're complex because they're made from living cells and they're not made from chemicals. And because they are made from living cells, they are naturally variable. They are produced by complex manufacturing processes. And more clinical comparative studies are required for a biosimilar's safety profile than for a generic drug. So a biosimilar must demonstrate it is highly biosimilar to its reference drug. So why biosimilars? And we're going to start with another question. So for question 2, biosimilar drugs are used in which of the following scenarios? So A, vaccines, B, blood and blood components, C, allergenics, D, gene therapies, E, recombinant therapeutic proteins, and F, all of the above. And I think we are all familiar with all of these categories having a biosimilar available for them. And so we'll talk about different examples over the next few slides. So biologic medicines have altered the way that severe, chronic, and disabling diseases are treated. And not only treated, but prevention of disease progression is another area where they're used. So examples would be that biologic medicines are highly specific. They're developed as targeted medicines, And they include the wide range of products that we saw in the question too. So vaccines, blood components, gene therapies, and tissues. And in the graphics on the right-hand side of the slide, you can see that they're used in oncology, rheumatology, gastroenterology, diabetes, dermatology. So they treat many fairly common conditions, but also some rare conditions. And they prevent disease progression as well as prevent severe diseases in multiple therapeutic areas. And so this has really revolutionized how medicine is delivered. And because of their efficacy and their cost, there really is a need for biosimilars. So the sales of biologic medicines in Canada is shown here on this bar graph from 2011 to 2020. And you can see on the left-hand side in 2011, $3 billion was invested in biosimilars in Canada. And over that decade, it tripled. So by 2020, we were at $10 billion. And that's a really important marker because with their strides in clinical medicine, we can anticipate that this is going to have further growth. So already into 2023, we're going to see higher outputs and sales of biological medications. When we look at availability of biosimilars to biologics, that has also increased. On this slide, what we're showing is information from Europe, the United States, and Canada. And each colored bar represents each region of the world. And what we're seeing is that in Europe, in particular in 2011, they had the highest availability of biosimilars and biologics. And then over the decade, that has increased pretty markedly in Europe, but also impressive uptake in North America, to the point that in 2020, you can see that Canada had higher use and uptake of it compared to the United States. And so this is an important thing, again, to recognize that it's becoming more commonplace and we need to anticipate that they will become available and that there may be some automatic switches that we're already seeing or that we can anticipate coming. And as more and more biologics lose their patent, we expect to see more biosimilars become available. So what is the rationale for using biosimilars? Really, the primary purpose of them is to decrease healthcare costs. And we know that biologic medicines are costly to manufacture. And the aim with decreasing their costs is also to increase access to healthcare so that if less expensive product is available, more people may have access to those medications. So the potential savings produced by biosimilar medicines can contribute to a more sustainable healthcare system. The magnitude of the cost differences and the financial incentives to utilize biosimilars by managed care and government payers can affect uptake of biosimilars in Canada. And in the next few slides, we'll see how these transitions are already ongoing in different provinces in Canada. So the costs of biological medications are rapidly growing. And in Canada, as well as globally, there's a lack of competition in the biologic space, which allows for increasing costs of biologic medicines. And so they're under patent for many years. And until they come off patent, biosimilars are really not manufactured and available. In 2020, biologic medicine sales in Canada were at about $10 billion with a 10-year compound annual growth rate of 13% in sales. And that was an increase of 15% in 2019 alone. And then other things that we need to consider are addressing the financial layout of biologic medicines, which has become a priority for provincial and federal governments, as well as private and public payers in different realms of the Canadian healthcare system. And so we're seeing that globally, biosimilars may potentially offer crucial cost savings to different healthcare systems while having the same clinical outcomes or similar clinical outcomes. So in the top left here, you can see that in Canada, in 2018, there was $94 million saved when we used biosimilars for these medications, etanercept, filgrastim, infliximab, and insulin glargine. So by using biosimilars of those four agents, we saved almost $100 million to the Canadian healthcare system. In 2019 alone, that was over $2 billion in the United States. And in other regions of the world that you can see on the right-hand side of the slide, we're talking upwards of $50 billion in Japan and $15 billion in Europe. So there are significant cost savings to using biosimilars compared to their original drugs. So as I mentioned, we do have different regions in Canada that are now automatically transitioning to biosimilar medications or switching to increase the uptake of these agents. And as of October 2022, we have this information available. So in terms of public payers, British Columbia, Alberta, New Brunswick, and Quebec, their patients are now required to use biosimilars of approved indications when they are available. And several other provinces, including Ontario, Manitoba, Nova Scotia, Newfoundland, the implementation of biosimilar switching is underway. And so we can expect to see more and more of this as time goes on. And then from a private sector, there are several insurance providers that are already initiating biosimilar transition programs for specific therapeutic categories of biologics. So for example, Sun Life and Green Shield Canada. Green Shield Canada has an asterisk listed here because it also allows patients to stay on their originator biologic, but the individual has to pay for the cost difference. And so that's a unique feature of that particular insurance provider. And so some factors that are impacting utilization of biosimilar agents, the uptake rates of biosimilars can be variable, and some of that has to do with the disease indication, especially if the indications are for life-threatening diseases or if there are life-threatening adverse events that may be associated with a particular agent, patient types, as well as physician specialty and their experience with reference biologics, lack of interchangeability, as well as whether or not you're using a biologic or a biosimilar for long-term maintenance therapy, or if it's only used once or twice, and then perceived potential implications of therapeutic non-response or adverse events. And when patient cost savings are not expected to be significant, some may wonder why use a biosimilar at all. So next we're looking at comparing a biologic to its biosimilars. And I'm going to pass this over to Dr. Gross to bring us through the next portion.
All right. So we're going to go through what biosimilars are based on their reference biologics. So biologic medicine, as we heard before, are produced in living organisms using cutting-edge technologies. They're large, complex molecules that can't be chemically synthesized. Because of this, they cannot be created or reproduced by conventional methods. They're essentially synthesized by a biologic system. So producing biologics, including biosimilars, is fundamentally more complicated than manufacturing a small chemically derived medicine. So what are the challenges in making a biologic or a biosimilar? So first of all, living cells are naturally variable. There's heterogeneity present between batches of biological medicines, and this will also apply to biosimilars. There's a degree, this degree of variability must fall within a range outlined by Health Canada. So in other words, the variation in a biologic exists, and the regulators know how to deal with that already. And so biosimilars essentially have to fall within that same variation. So there's strict manufacturing controls to ensure batch-to-batch consistency. So this graphic sort of goes through a complex molecule being processed that is then analyzed. Health Canada looks at it, and only if it's the same is it approved or continued to be marketed. So during a life cycle, existing biological medicines have changes in how they're made. So there could be an advance in their process, like a technological advance, like low liquid heparins were, started to be made in the late 80s, early 90s, and I'm sure factories and how they're made and how they're processed, et cetera, has changed. The yield and strain of the cells can be improved upon. You can have new purification methods. Cell lines shift and change and can be modified. And factories can change. You can have a shift in manufacturing sites. So each time one of these changes occur, there's comparability testing that has to happen to ensure safety and efficacy is maintained. And this is across different versions of the same biologic medicine. So here's a figure from a paper looking at approved manufacturing changes for monoclonal antibodies according to their risk category. The majority were low and moderate risk changes, but a few were high risk, potential high risk changes that had to be ensure that had to be checked to ensure safety and efficacy. So reference biological medicine manufacturing changes are approved after a stepwise comparability exercise. So the originator, the original drug manufacturer, has to use analytics and projections of indications to gain approval for the process alterations. Then the regulators have expertise evaluating and approving changes, meeting internationally agreed standards for how to compare these. Once comparability is shown, the new version is launched. So this is just what's in existence for biologicals, never mind biosimilars. So the scientific principles for establishing biosimilarity are essentially the same as this for demonstrating a change in the process of an already licensed biological medicine. So here's the take-home point that you need to know. Biosimilar must be within 20% of the biologic, the reference biologic, in various criteria to be approved by Health Canada. So within 20% is sort of the rule. So a question. So the concept of totality of evidence required when validating a biosimilar compared to its reference drug does not require which of the following aspects: proof that the biosimilar is manufactured in the same regulatory standards, that the biosimilar is established to be highly similar, that the biosimilar exhibited no clinically meaningful differences in efficacy and safety in comparison to its reference drug, and D, that a phase 3 trial with large numbers showing efficacy and safety exists. So here's where we hate this. And the wrong answer is D, okay? So the safety and efficacy of the biological reference product has already been proven. So biosimilars do not need a phase 3 trial to get approval. They need the other factors. So what does totality of evidence mean? So the Health Canada regulatory review and approval, these things are under acts, the Food and Drug Act and the Food and Drug Regulations. The manufacturer provides totality of evidence validating that the biosimilar compound is compared to the reference drug. It has to be manufactured to the same regulatory standards. It has to be established to be highly similar, at 20%. It has to be exhibited. It has exhibited no clinically meaningful differences in efficacy and safety in comparison to the reference drug. And then Health Canada will authorize a biosimilar once all these steps are completed. So the regulatory and development stepwise process for a biosimilar is kind of upside down compared to the reference product, okay? So when you start with a reference product, you have the chemical synthesis and analysis, the preclinical work, the phase 1, and then phase 2, and then phase 3 trials. When you have a biosimilar, it's sort of the triangle's upside down. You do that analytical work to make sure that what you made is the same. Preclinical work to make sure that it is the same and it has the same effects. And then a phase 1 PK/PD pharmacokinetic pharmacodynamic study to show that it is similar to the reference drug. And then a confirmatory clinical phase 2 is usually all that's done. So the biosimilar drugs must establish high similarity to the reference biologic within all these criteria. PKD profile of the biosimilar must be scrutinized closely, again, within this 20%. So the key part of biosimilar medicine development is the PK/PD comparability, which establishes that the biodistribution is similar to the reference drug. So PK is concentration over time, and PD is concentration versus effect. And then you get the effect first time in the PK/PD graph. This is a crucial indicator of the clinical biosimilarity between the biosimilar and the reference drug. And comparative analytic non-clinical and human PK/PD, including immunogenicity studies, have to show this biosimilarity. PKD biomarkers allow for shorter and less costly clinical studies. However, special patient populations may require further biosimilarity studies, and this is up to Health Canada. So the extrapolation of the evidence is the totality of evidence concept. Okay, there's no one size fits all. It's a combination of all these things submitted by the biosimilar company as evidence. The analytical characterization of the overall information comparing the biosimilar to the reference biologic, the safety and efficacy of the reference product has already been proven, so you just need to say that you're similar to that. Health Canada scientists will evaluate these in totality and integrate all the information and provide an overall assessment of whether the biosimilar is highly similar to the reference product, and if it is, it's approved. A summary of the requirements is the manufacturers have to demonstrate the similarity of their product to the Canadian reference product by using comparative studies, as we've shown. The type of data required to support a biosimilar authorization differs from the data for a standalone biologic drug or a new drug submission. The manufacturer has to independently demonstrate the biosimilar against previously authorized reference product. They have to demonstrate the quality of the biosimilar drug, perform comparative studies to demonstrate its similarity in structure, its similarity in function, its similarity in efficacy, and its similarity in safety. And PKD studies are the most important when it comes to this. So now we're going to switch to biosimilars and thrombosis in specific. So let's have a patient, Etienne, 67-year-old man with metastatic lung carcinoma. His course is complicated by segmental right upper lobe pulmonary embolism, and he's currently on a direct oral anticoagulant. You're seeing him because he's developed breakthrough deep vein thrombosis in his right leg and progressive pulmonary embolism while on the direct oral anticoagulant. So would a biosimilar low-molecular heparin be appropriate in this context? So we're not going to answer that. We just want you to think about it as we go through the rest of the talk. So the characteristics of the originator and biosimilar low-molecular heparins. For the originator reference low-molecular heparins, inhibition of anti-10A and antithrombin varies on the manufacturing methods, the chemical profiles, and the unique fragment sizes of the different products. Biosimilars are required to be highly similar, not identical, and to be approved by Health Canada as being biosimilar. No clinically relevant differences are to be expected. The originator low-molecular heparin the originator low-molecular heparin is also different in the proportion of fragments per milligram, which inhibit 10A and thrombin, the specificity and therefore the impact of the amount of non-anticoagulant fragments. The Health Canada requirements are PK/PD studies. However, not all biosimilars have phase 3 clinical trials in patients with disease, although there are some that do. So are low-liquid heparins, even the comparators, interchangeable? And the answer is they're not. Not all low-liquid heparins are the same. One low-liquid heparin cannot be interchanged with another low-liquid heparin because they differ in manufacturing process, they differ in molecular weight distribution, they differ in their anti-10A and anti-thrombin activities, they differ in the units and the dosages, and they differ in the raw material, the animal cells that manufacture them. So just to go through in this chart, you can see that the mean molecular weight, and again, this is a distribution of molecular weights of the different low-liquid heparins in Canada compared with unfractionated heparin are different. The anti-10A to anti-thrombin ratio is as high as 3.8 with enoxaparin and as low as 1.9 with tinzaparin. The reported half-lives are also different. And lastly, how the heparin is cleaved into low-liquid heparin fragments is different. Chemical cleavage using alkaline reaction for dalteparin, by nitrous acid for enoxaparin and narteparin, and by enzymatic depolymerization for tinzaparin. So focusing a little bit more on enoxaparin because it has biosimilars. Enoxaparin and its biosimilars are approved for the same indications. Biosimilars to low-liquid heparin in Canada have been available since the spring of 2021. Health Canada approved 3 biosimilars for the same indications as enoxaparin, brand name Lovenox. These are Inclinox and Inclinox HP, Narombi and Narombi HP, and Redesca HP. On the right are the indications for enoxaparin, and these are the same indications for the 3 biosimilars. So prophylaxis of DVT, prophylaxis of DVT in medical patients, top is in surgical patients, prevention of thrombus formation in extracorporeal circulation during hemodialysis, treatment of DVT with or without PE, the treatment of unstable angina or non-Q wave MIs with aspirin, the treatment of acute ST elevation MIs, including patients to be managed medically or with subsequent PCI. So what do we know about these 3 biosimilars? Redesca stands a little bit different from the other 2 because it is vertically integrated from a secure heparin source. So heparins are isolated from animal intestines, and this largely comes from China for most of the products. The company that isolates the raw heparin is the company that makes the Redesca. They sell the raw product to other companies that manufacture the other low-liquid heparins, some of the other low-liquid heparins. There's worldwide experience of the 3 drugs a little bit longer for Redesca, over 10 years. Redesca has a small phase 3 comparison in patients with high-risk DVT. The pharmacodynamic study was used with 100 milligrams for Inclinox and Narombi and 40 milligrams for Redesca. There's been a small post-marketing safety study in Europe with Redesca, but not the other 2. All 3 of the drugs use the Preventus active safety device to shield the sharp needle. So thinking back to Etienne, I'll just bring this up again, although we haven't really given you a lot of information about how you would come up with an answer to this, but something to think about. So questions about low-liquid heparin biosimilars? we'll go through these questions. Why is not every indication of a reference drug studied for a biosimilar? Why there's so few phase 3 trials? From where are the active pharmacological ingredients and the source of the heparin derived? Are low-liquid heparins biosimilars safe for my patients? So why is there not every indication for the reference drug studied for a biosimilar? So indications authorized for biosimilar depend on some factors. Some indications may be patented at different times. And so the indications for the intellectual property protection cannot be authorized until the protection expires. So you can't have a biosimilar if the reference drug is still under intellectual property protection for that indication. The biosimilar manufacturer can choose which indications to apply for. Health Canada doesn't force the manufacturers to apply for specific indications. They can apply for whichever indications they wish to. So the Health Canada summary basis decision, which is essentially the approval, gives details on which indications the biosimilar sponsor did and did not seek authorization. So why are there so few phase 3 trials for the biosimilars? So Health Canada doesn't always require a biosimilar phase 3 comparative trial carried out in patients being treated, because they have extensive structural and functional studies that are used to deliver the totality of evidence for authorization of the biosimilar. So as we've said before, the evidence includes studies that compare product stability, biological activity, physiochemical properties, immunochemical properties, purity and impurity profiles. Comparative studies are considered more sensitive than clinical studies to detect clinical meaningful differences. So this is something that, as clinicians who practice evidence-based medicine, we have the hardest thing accepting that middle box. The purpose of human clinical studies is to show that there's no clinically meaningful differences in efficacy and safety. So are biosimilars safe for my patients? So some points to consider. Different manufacturing processes result in unique chemical structures. Heparins and low-liquid heparins should not be used interchangeably with other low-liquid heparins or unfractionated heparin. I often talk about low-liquid heparins as chicken soup. My mother makes very good chicken soup, but it's not the same every Friday, okay? The recipe is about the same, but it's not the same. And your mother might make chicken soup and it'll be chicken soup, but it might not be my mother's chicken soup, okay? Pharmacovigilance surveillance programs also exist, which depend on reporting by healthcare practitioners. One report will not identify an association, but many reports with a similar history are needed to produce a meaningful association, recognition, and action. There was concern raised by an Italian thrombosis group, CISET-CIAPAV. I won't make people laugh on my poor Italian of what those abbreviations mean in 2017. This is published by Imberti in the Thrombosis Journal. You can see the reference at the bottom. Regarding the European Medical Association's process for approval of enoxaparin biosimilars. They stated in the paper, regarding the enoxaparin biosimilars, actual clinical similarity to the originator reference drug. They say the data could not draw proper conclusions regarding the safety profile of the drug. The assessment of the safety was based on small-scale PK/PD studies that relied on small, underpowered, healthy volunteer study. This is what is needed by the regulators to approve a biosimilar. Recommendations included promoting well-designed and powered studies, and they say this will aim to establish actual efficacy and safety of biosimilars. So other points to consider. There's a large body of confirmatory evidence combining 13 plus years of European biosimilar medicines clinical use. Over 700 million patients have received biosimilars. there's a quote from the European Medicines. Over the last 10 years, the EU monitoring system for safety concerns has not identified any difference in the nature, severity, or frequency of adverse events between biosimilars and their reference medicine. There's one trial, one controlled trial involved switching people from infliximab to its biosimilar called the North Switch trial. So the last patient not the last patient, another patient we'll talk about. There's going to be one more patient. We chose this patient because I think it's a little bit more to wake you up and have you thinking of who you would think about a biosimilar for. So Michelle's a 42-year-old self-employed social media consultant. She has newly diagnosed gastric adenocarcinoma in situ. She develops a new deep vein thrombosis. She has no drug coverage. She's not been exposed to an originator, and she cannot afford the reference drug. She has no insurance. So what options for anticoagulation can you offer her, being mindful of her financial limitations? So I think, this is, when thinking about a case like this, is where you really start to sort of think about, and it's tailored and it's quite specific, where you might think about, where your first foray into biosimilars might be. I'll hand it over to Lana. You should be good.
All right, thank you, Peter. Okay, so next we're going to talk about how do we discuss biosimilars with patients and their family and caregivers. So we'll talk about this patient, Achak, who's an 81-year-old gentleman that recently experienced an MI, and he's presenting to the pharmacist with a prescription for enoxaparin. And the pharmacist mentions that he's being given a biosimilar medication. And Achak wants to know what that means. So we're going to talk about some terms and phrases that we can use to explain biosimilars to patients. So when we talk about biosimilars with our patients, there are some key points and highlights that we want to share. And we really want to emphasize that biosimilars are similar with the overall effects and similar side effect profiles to the original drugs, and that they are reviewed by Health Canada and must demonstrate that they have no clinically meaningful difference compared to the original biologic drugs. And the language that we use when sharing this information with patients and family members is also important. So what often comes up is that biosimilars are intended to save healthcare costs, but only if successfully initiated or switched. And so it's important to talk to patients about wanting sustainable healthcare system, access to medications for all patients, and using biosimilars may help to provide better access to more people to certain medications and therapies. It's also important to initiate a discussion on transitions that are already happening. So as we saw earlier in the presentation, several provinces have already undergone automatic switching programs, and other provinces are in the process of coming up with those strategies. And so this is an important thing to recognize and appreciate, that it's inevitable that a lot of this switching will already be happening in the background and won't necessarily be under our control or a patient's control. And it's also important to identify which patients may be appropriate for switching. So take note of the different instructions for self-injection when teaching patients about self-injection of low molecular weight heparins, for example. So the different administration devices, whether there's pre-filled syringes that are spring-loaded or not, it's important to be able to teach patients different strategies for using those different device mechanisms. And then also important to have a good relationship with your pharmacist, both in the hospital as well as in the community, where a lot of these switches may be happening even without our knowledge. So they may automatically be doing these changes and providing biosimilars to patients. So when we look at the switch, scheduling a specific discussion time with your patient about the reason for switching or the possibility of switching back to their reference agent may be something that needs to happen. And this is, again, done to reinforce the trust and relationship that you as a healthcare professional have with your patient. Sending out information letters to patients with the proposed switch that may be coming, or whether it's an automatic switch or an anticipated switch voluntarily in the coming months or years. And, you know, when we do look at an overall general sense from healthcare professionals, there is not necessarily agreement about automatic substitution of biosimilars at the pharmacy level. So some people do continue to have reservations about an automatic substitution. And that's another thing that we need to be aware of, that not everybody's in agreement. And we'll get to some of the questions in the chat, but there's some good comments in there about is that 20% similarity enough? And so those are a lot of the lingering questions that many of us still have. So to summarize for today's presentation, when we're talking about low molecular weight heparin biosimilars, as we recognize and appreciate, not all low molecular weight heparins are interchangeable, and they differ in manufacturing process, their molecular weight distribution, their anti-10A and thrombin activities, their units and dosages, as well as the source of raw material. All three Health Canada approved biosimilars have the same indications as enoxaparin. And over 10 years of European biosimilar clinical use in over 700 million patients is reassuring that no differences in safety between biosimilars and their biologic originator drug exist. And biosimilars can save healthcare costs. So as I mentioned earlier, please use the chat feature for your questions. And Peter and I will do our best to answer questions. And as I mentioned, there's some interesting comments that are available in the chat as well, that you know, talking about whether or not the 20% margin for a biosimilar compared to the originator drug is appropriate. And Peter, I don't know if you have a comment on that or a thought about that.
Sure. So I think, you know, you have to understand that 20% in the proper way of what it is, right? So it doesn't mean that it's definitely 20% better or 20% worse. All these PK and PD parameters have error bars, right? So you have to, the confidence interval of the point estimate has to exclude 0.8 and 1.2 of what the reference interval of what the reference drug is. So it's not, we're not saying that it's going to be 80%, as bad as 80% or as bad as 120%. It has to exclude being as bad as 80% and exclude being as bad as 120% of the reference drug. So I think that's sort of a key thing. And also, you know, low liquid heparins are variable, right? So I think if you had in your drawer some preserved enoxaparin from 15 years ago and brand new enoxaparin from today, it's probably going to be again the same within 20% of it. That's the problem that we've already been dealt with. We already deal with variation in low molecular weight heparin. So I think this is just another version of variation.
Thank you, Peter. That's great. And some other comments about specific low molecular weight heparins that we're referring to. Thank you, Dr. Agnes Lee, for highlighting that. We are specifically talking about biosimilars to enoxaparin. We are not referring to any biosimilars for dalteparin or tinzaparin, as those are not yet available. So this presentation is specifically targeting enoxaparin biosimilars. And to finish Aggie's question, sorry to interrupt. That's okay.
And so if you are uncomfortable using enoxaparin for a situation, then you would be uncomfortable using the biosimilar, right? Absolutely right. So if you're uncomfortable using enoxaparin in Dr. Lee's favorite topic, cancer-associated thrombosis, because frankly, I don't know what dose to use, then I would be uncomfortable using a biosimilar in that situation. There is some stuff that's weak evidence about the once daily 1.5 milligram per kilogram, but I'm not sure that is the phase 3 evidence we would like to have to justify that. But absolutely agree.
Great, thank you, Peter. And just some comments about availability in terms of cost coverage for the biosimilars compared to Lovenox. And I think this is certainly dependent on where you live in Canada, different provincial sources for funding. For example, in Ontario, Redesca is covered under Ontario drug benefits for patients. And in other provinces, there may not be the same reimbursement strategy. So I think that depends on where you live, and working with your local pharmaceutical representative to see what coverage is available for cost assistance for patients is useful.
So to answer Jennifer Yun's question, there's safety data about biosimilar enoxaparins for DVT prophylaxis, but not for therapeutic doses. So Health Canada doesn't care about that. If the PK of the prophylactic dose was biosimilar to the reference product, then they would assume, given that the structure is also biosimilar and the molecular weight is biosimilar and everything else submitted by the companies says that it's biosimilar, that the therapeutic dose would also be biosimilar. And that is enough for the totality of evidence. So in other words, there is no phase 3 trial of DVT treatment of biosimilar enoxaparins versus enoxaparin. And you would need thousands of people to prove that.
Yeah, that's a great point. Difficult to do these trials already, and then to do them again a second time would be even more challenging in some ways. So thank you, Peter, for answering that. There's some ongoing discussions in the chat about whether for our first case for Etienne, the patient with lung cancer, whether or not a biosimilar would be used. And I think as Aggie and Peter mentioned, if you wouldn't use enoxaparin for that patient, then a biosimilar likely doesn't have a replacement for it. So I think you would use your best clinical judgment in that scenario, as with our other scenarios as well, the other 2 patient cases that we had discussed. Some questions that are pretty relevant here in terms of concerns with using a biosimilar in the pregnancy population. Personally, I tend to use more dalteparin for pregnancy-associated thrombosis and or prevention, and mainly because that's where the clinical trials have supported that. I don't necessarily use the low molecular weight heparins interchangeably in that way for prevention or treatment. And so we're likely not going to have a robust amount of data in the pregnancy population with biosimilars. But again, if you wouldn't use enoxaparin in your pregnant patient population, then consider avoiding using a biosimilar in that population as well.
Someone asked about checking anti-10A levels after switching to a biosimilar. I don't do anti-10A levels for any reason. I certainly wouldn't do it if I prescribed a biosimilar.
Fair enough. And then Agnes is asking if the biosimilar company is providing samples or teaching materials. And I can say that yes, in Ontario, they have done both of those things for us, at least in our Ottawa clinic. I don't know about other regions in Canada, but again, I think important to make that connection. And somebody is saying that yes, in Alberta, they have them, and as well as in Hamilton. So I think it depends on the region you're in.
There are quite a few questions about Etienne, the person with the breakthrough DVT and PE on a DOAC. And, I think that's sort of debatable therapeutic choice. And if you put together cancer thrombosis experts from around the world, you could get different answers about what to do. So, I'm not sure that I would put someone like that on enoxaparin of any kind. as I don't know what dose that I would give them. So like, I'm not sure, like, so the question is, would I use a biosimilar for that patient? No, but I wouldn't use Lovenox either. But others may have other answers.
Mm-hmm. No, absolutely. So thank you for participating today and for your great questions and comments. I also want to highlight that we have a unique conference experience and opportunity happening in October 2023. It's the Vascular 2023 conference in Montreal. And this is a grouping of six different organizations across Canada that will be having their conferences during this week, and it includes CCS, Diabetes Canada, Thrombosis Canada, CanVector, our national research organization, and a few other organizations. And so this is a great opportunity to consolidate a lot of your knowledge and learning and networking opportunities into that week. So save the date, and we hope to see many of you there. Thanks again for your participation today. And on behalf of myself and Peter and Thrombosis Canada, thank you, and we wish you all a good day.